Welcome to this lecture on cell biology. Today, we are going to explore one of the most fascinating and crucial structures in our bodies, the adherens junction. We will discover how microscopic molecular belts keep our cells firmly anchored together, allowing our tissues to withstand incredible physical stress without tearing apart. To understand why adherens junctions matter, imagine you and a friend are wearing heavy-duty belts linked together with strong metal carabiners. If someone tries to pull you apart, those belts distribute the pulling force across your entire waists, protecting your bodies from localized strain. In the microscopic world, tissues like our skin and beating heart experience constant stretching. Cells need a very similar mechanism to stay attached. Adherens junctions act exactly like these linked belts, ensuring that when a tissue is pulled, the cells do not simply rip apart from each other. Now that we understand the mechanical challenge, let us look at the biological solution. Formally, an adherens junction is a specialized cell-to-cell junction that provides strong mechanical attachments. The primary building blocks of this structure are transmembrane proteins called cadherins. You can think of cadherins as the hooks reaching out from the cell surface. They bridge the tiny extracellular gap between two adjacent cells by binding directly to identical cadherins on the neighboring cell. This homophilic binding creates the initial physical connection between the two cells. This initial connection brings us to the inside of the cell. If cadherins were just floating freely in the cell membrane, they would easily be ripped out under stress. Instead, on the inside of the cell, these cadherins are firmly anchored to a dense plaque of intracellular proteins known as catenins. These catenins act as a crucial adapter, binding directly to the actin cytoskeleton, which is the internal structural framework of the cell. By connecting the exterior cadherins to the interior actin filaments, adherens junctions create a continuous, highly resilient mechanical network across an entire sheet of cells. Before we move on to the biophysics, let us see this structure visually. In this diagram, you are looking at the boundary between two adjacent cells. The vertical lines represent the plasma membranes. In the extracellular space in the middle, you can see the cadherin proteins, interlocking, much like Velcro. Notice how these cadherins pass through the membrane and connect to the catenins, shown as the dense plaques on the inside. Finally, follow the catenins to the thick actin filaments extending into the cell's interior. This diagram perfectly illustrates how external forces are transformed deep into the cell's structural framework. Having seen the structure, we must ask what happens when it fails. Without adherens junctions, our bodies would literally fall apart under the slightest physical stress. Tissues like the epithelial layer of our skin rely heavily on these junctions. If a genetic mutation causes a defect in either cadherins or catenins, the tissues become incredibly fragile, leading to severe blistering diseases. Furthermore, in cancer metastasis, tumor cells often downregulate or turn off their adherens junctions. This loss of adhesion allows them to break free from their original tissue and travel dangerously to other parts of the body. This biological reality leads us to a biophysical formula. We can actually calculate the total mechanical force required to tear two cells apart, denoted as F total. This force depends on three variables. First, ρ represents the density of cadherin proteins on the membrane. Second, A is the total surface area of the adherens junction. Finally, F bond is the microscopic force required to break just one single cadherin to cadherin bond. Conceptually, this formula tells us that the total strength depends on how densely packed the Velcro hooks are, how large the patch of Velcro is, and how strong each individual hook is. If the density increases, the total adhesive force increases. Now that we have our formula, let us see this with a concrete example. We are given an adherence junction with an area of 2.0 square micrometers, a cadherin density of 500 proteins per square micrometer, and a single bond strength of 40 times 10 to the negative 12 newtons. We need to find the total adhesive force. First, we calculate the total number of individual bonds by multiplying the density by the area, giving us exactly 1,000 active bonds. Next, we multiply these 1,000 bonds by the force required to break a single bond. This gives us a final answer of 4.0 times 10 to the negative 8 newtons, or 40 nanonewtons. This shows how thousands of weak molecular bonds combine to create massive cellular strength. Building on that calculation, let us explore the effect of cancer on tissue strength. First, we must establish the baseline mechanical strength of a healthy tissue. We are given a junction area of 3.0 square micrometers and a healthy cadherin density of 600 proteins per square micrometer. The bond strength remains 40 times 10 to the negative 12 newtons. By multiplying the healthy density, the junction area, and the individual bond strength together, we find the normal adhesive force. The result is 7.2 times 10 to the negative 8 newtons. This value represents the strong, reliable anchor, keeping the normal cell firmly in its proper place. Now, let us see what happens when a cancerous mutation occurs. The mutation causes the cell to downregulate cadherin expression, dropping the density to just 150 proteins per square micrometer. The area and bond strength remain exactly the same. We substitute this new, lower density into our formula. Because the density dropped so drastically, the total number of active bonds is severely reduced. Multiplying the new density by the area and bond strength gives us a new adhesive force of only 1.8 times 10 to the negative 8th newtons. This mathematical drop perfectly mirrors the biological reality of a cell losing its grip on its neighbors. To truly understand the severity of this mutation, we need to compare the healthy and cancerous states. We do this using the standard percentage decrease formula. The formula takes the difference between the healthy force and the weaker cancer force and divides that difference by the original healthy force to find the proportional loss. Finally, we multiply that decimal by 100 to convert it into a readable percentage. This mathematical tool helps us quantify exactly how much structural integrity the tissue has lost due to the downregulation of cadherins, giving us a clear picture of the disease's impact. Let us plug our values into this formula to find the exact loss. We take our healthy force of 7.2 times 10 to the negative 8 and subtract the cancer force of 1.8 times 10 to the negative 8. We then divide that difference by the healthy force. The math simplifies beautifully to 0.75. Multiplying by 100 gives us a final answer of a 75% decrease. Practically, this means the cancer cell has lost 3/4 of its physical attachment. It is barely holding on, which perfectly explains why malignant tumors can easily shed cells into the bloodstream, leading to metastasis. We have covered a lot of ground today, so let us summarize the key takeaways. First, Adherens junctions are essential mechanical fasteners that lock adjacent cells together, allowing tissues to withstand physical stress. Second, they rely on a brilliant tripartite structure: transmembrane cadherins linking externally, anchored by catenines to the strong actin cytoskeleton internally. Third, the total adhesive force is a direct product of cadherin density, junction area, and individual bond strength. Finally, understanding this biophysics explains why the loss of these junctions leads to severe blistering diseases and enables cancer metastasis. Excellent work following along with both the biology and the math today.